live here. I'm going to turn on recording, and after that we can start admitting the people. I was recording. Stop recording. Resume recording. Okay. All right. Perfect. Thank you very much for joining. As we're waiting for a couple of people, we have a quick poll. It should be very simple. When do we think the pandemic will end? Is the question on everybody's mind.
The question is just not fair.
It will never. Yeah, I think it's a good measurement of the people optimism or pessimism. So sort of kind of depends.
Maybe realist.
Well, what about pragmatism? I mean,
I don't know. Well, I don't think so. We can solve the pandemic, but the one thing, and this is our results from the group, so that you guys can take a quick look. But I think interesting thing, we can start what we came here for, which means we can open the session of the Society of Healthcare Innovation and University of California, Santa Cruz. Today we have an exciting event. My name is Chris Radomski. I'm the president of the SHCI, and I'm proud to announce our last session of the Frontiers 2021. What we're going to talk today is actually CRISPR and the biohacking, and the need for that particular session emerged after discussion with our constituents in the organization and with the people at the University of California, Santa Cruz, when we're discussing how easy it's now to do gene editing and actually doing anything with the gene. Almost, we kind of joke that this may be done in the garage, and I thought that I'm joking about it until I met Raymond and Samantha, and I learned that actually you can do this in the garage if you really, really push. So this is the subject of today's session. I'm going now to hand the reins over to Dr. Camps, who will introduce moderators. Dr. Camps.
Hey, Chris. This is my pleasure to introduce the two moderators for this session, and also the two organizations that co-organized this session. So the first moderator you've already met, it's Chris Laudansky. So he is an MD, PhD in MHCI, an assistant professor in the Department of Anesthesiology and Critical Care at the University of Pennsylvania. And he leads this organization, and he's been instrumental for setting up this event. The second moderator is David Bernick. So he is a teaching professor at the University of Santa Cruz, a mentor and a bioengineer. He works on projects that strive to solve issues of global significance where engineering of a biosystem can yield an important benefit to society. He teaches courses in programming, algorithms, and bioinformatics, an integrated course field to journal in extreme virology, and a seminar in team skills for bioengineers. David directs the iGEM undergraduate research series as well. And each of these courses takes on a hands-on approach where students build, discover, and present their work. The two organizations that are organizing this event are SHCI. So SHCI is the Society for Healthcare Innovation. It was founded in 2020 and by a group of healthcare professionals interested in finding solutions for healthcare delivery using new technologies. It strives to create a community of healthcare innovators, promoting educational awareness and the professional development of their members. Their goal is really to empower the members to create new and valuable healthcare solutions. The second organizer is Crown College at the University of California in Santa Cruz. Crown College is one of the 10 colleges of the university, and our theme is examining the role or the impact of technology in society. Crown also sponsors two organizations. So one of them is the Center for Applied Ethics and Values of Emerging Technologies. And the other one is Get Virtual, which uses technology to assist local businesses. So without any further ado, please enjoy this conference.
Thank you very much. So how the conference will go is in front of you. So we just started. This is our introduction. After that, we're going to have two presentations side by side by our esteemed speakers. This will be followed by Q&A from the moderators. Sometimes we have a break room, sometimes we don't have a break rooms, but we always take the questions from our audience. So please post anything in the chat. You can raise your hands to let us know that you post something, but the best way is to communicate with us is via chat. And around 3:30, we're going to close. And sometimes we ask for somebody engaged to stay longer to meet with us or the speakers. Couple details about the behavior. We all live by Zoom these days, but just please remember, mute your audio. Sometimes, you know, this silence button press one too few kind of creates a lot of havoc in your life. Make sure that the camera is positioned properly, especially when you look at the background. I steal the decoration idea, home decor idea from the people by looking at the Zoom. So be aware of that. When speaking, be mindful of the background noise. If we go into the breakout room, we have a special guidelines for that. And then please remember that all conference sessions are recorded and will be available to everybody who registered in our constituents. Now, let's move to our speaker. And David, if you can introduce them, would be fantastic.
Yeah, I'm very pleased to introduce Samantha Mirage. Samantha comes to us from, specializes in human genetics and molecular biology from Johns Hopkins. And she leads the genome editing program at NIST, and which has a primary focus on measurements and assay applications in genome editing. And I'll turn it over to you, Samantha.
Actually, David, if you could do me a favor and also talk about the Raymond, that would be great.
Oh, excellent, excellent. So Raymond Macaulay, so he is going to be speaking second today. He's co-founder of BioCurious, which is a hackerspace for biotech. Also is a principal of exponential biosciences and has done prior work with next generation sequencing at Illumina. And today he'll be talking about a focus on CRISPR and DIY.
Perfect. So now we're going to turn this over to Samantha. Samantha, if you can share the slide, would be fantastic.
Great. All right, you should be seeing my screen and you should now be seeing it in presenter mode.
We do. Thank you.
All good.
Wonderful. All right. So thank you all for having me here today. I want to take you on a very quick whirlwind on a bit about genome editing and introduction and some of the things that we're doing at NIST to help move this technology towards products that you might be able to engage with. So we're talking about CRISPR today. I want you to see down here in this window. Let's see if I can get my laser pointer here for you. So CRISPR-Cas is right here on the system. And what we want to do is give you a sense of how somebody might be using a CRISPR system. So one of the things I just want to note is that while CRISPR is fantastically flexible and a really accessible technology to people, there are some other technologies that are a little bit more challenging to use in a non-standard lab that has a lot of setup space. CRISPR has brought this far down, very far to where you don't need as many technical expertise to be able to make these molecules and potentially use them. And there are many, 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 many labs across the world that are using this technology. But I just want to make sure that you note that there are also other technologies that are being used to do similar sorts of things. So I'll call this broadly genome editing molecules and genome editing systems. So you have these genome editing systems, and what you're going to do is you're going to design them such that they will be complementary to a sequence of DNA, or in some systems RNA, that you want to change. And so you design that first, and then you have to figure out how you're going to get these systems into the cells that you want to change. Because one of the great things about this is that you're actually able to change the DNA sequence of live cells while they're still alive without killing them, and then have these cells be functional and hopefully have a DNA sequence change that's meaningful to you, so that these cells are alive, functional, but now have a different sequence And that sequence should hopefully give some new capability that you would like it to have. And so then you have to figure out, do the cells do what you want them to do? And the kinds of things that you might want to do with them is they might be what I call tools to learn, they might be tools to make, or they might be your final end products. Okay, so examples of tools to learn. There's several things here in terms of the more applicable space to maybe humans. Then a lot of times we think about these systems as, engineering cells so that they can tell us something about the function of the cell. And so the biggest thing that I think about in my world as somebody that's in human genetics and molecular biology is reporter cell lines. You might put fluorophores in or something else that you might be able to visually see, tell you something about the cell's function. But these aren't limited to mammalian cells or eukaryotic cells. People are even using these in microbes. So when we talk about examples of tools to make, you might be engineering these cells so that they are helping to make you some secondary product. So in this example here, we have sort of traditional production of some sort of small molecule drug. And that's usually some biochemical or some chemical factory that's making this product. But you might engineer, say, bacteria to be able to make that same product, and you might be able to make it even more efficiently than you're doing that now. And that's a really big application in this space. And then the application that I'm closest to is this end product space. So where the cells that you're engineering are actually the thing that you want to have interact with the public in some way. That might be therapeutics. I'll give you a little bit more about that. Here's another example where engineering some sort of organism. Mosquitoes is one particular example where you're engineering them so that you're remediating their ability to cause or carry disease. Maybe targeting microbes to eliminate antibiotic or antiviral resistance. It might be something in agriculture like plants or livestock that you're engineering so that they can be more robust for production, or other kinds of treatments for healthcare. So for this gene therapy use case, just to center everybody, there are two big kind of examples where it might be autologous, might be taking cells from a patient, engineering them with, say, CRISPR, and then trying to put those cells back into a patient. Maybe you've got a healthy person and they're donating cells, and they can be engineered so those healthy cells can actually be a therapy for other people and other patients. And maybe you're trying to deliver those CRISPR systems directly to a patient. And the reason that this might be really important to think about is that not everything that you want to engineer, you actually have the ability to take out of a person. So you can't take their heart out, you can't take their brain out. And maybe this is actually the only option you have to get this person a therapy that reaches the tissues that you need them to get to. So you can think of all these broadly, we call this ex vivo, and broadly we call this in vivo. All right, so what's the big deal here? Why can't we just do this? Genome editing outcomes aren't always what you think they are. So this is the challenge that my area at NIST that deals with standards and norms tries to think about. Is that when you think about genome editing, when you think about applying a CRISPR system, you have something that you're trying to change, right? Some DNA sequence, if we're talking about, say, the human genome, that you want to change, and we call that your target. So when you apply these molecules, you may get as the output an edit that you want. So the big thing here is that, say, a CRISPR-Cas system, we call it genome editing, we call it editing, but what it actually is doing is it's generating a DNA break. It doesn't have the ability to tell the cell how to fix the break, what sequence to put there. You can try things to bias that, but by its nature, all it's doing is making a DNA double-strand break. And so it's actually more accurate to characterize it as like a DNA damaging system. And what you hope is that you can use some techniques that allow you to get a DNA sequence change when the cell repairs that break, because cells do not like to have broken DNA, they're going to repair it, that you get some DNA sequence change that's meaningful for you, that's fit for your purpose, whatever you're intending. You could get that at your target, you could get no edits, or you could get what we call unintended edits, which are DNA sequence changes that you don't mean to happen, that can happen even at your target. Okay, so that's at your target, which might be one or a very few number of positions in the genome. But then what about the rest of the genome? Well, these systems aren't perfect. So at some frequency, they have the ability to cut or damage the DNA in places you don't mean for that to happen. And whenever you create those breaks, there's the possibility that you can have an edit. And so anywhere that's not your target, that you generate these DNA sequence changes because you applied CRISPR to your system, we call these unintended edits. And we call anywhere that's not your target an off target. So the real thing is that this happens. Off targets happen. They're difficult to measure, but they're important to be able to measure to understand what else did you do to the cells that you didn't need to do, and does it matter? If you're going towards a therapeutic application, the FDA requires you to tell them about this because they want to know, is this thing safe? You did this, yes, maybe you did this thing that you meant to do, but what else did you do that you didn't mean to do? And is that concerning that that's actually there? So when I say genome editing outcomes are complex, I want you to give, here's a real example I wanted to give you. So here's how your workflow could potentially go. You have some cells. This could be example of cells in a dish, like here, this could be the cells in somebody's body. You apply your genome editing molecules, you either inject or you get some way of getting these molecules into the cells. And let's just only consider this as your target. You might have unedited cells. You might have cells that have your desired edits. You might have cells that have undesired edits, right? And when you do this, these are all together, right? You very rarely have the opportunity to actually clone and get only the cell that you want and use that as your product. So this is real data of an editing experiment where all of these are happening at the same time because we're taking a subsample of this pool. And I'm not going to read through these common challenges, but there are lots of challenges with understanding, even at your target, what you've done and how sure you are that you've understood what you've done. This can be overcome by very deep targeted sequencing. For small sequence changes, for very large sequence changes, you need different approaches, different techniques to understand what's done. And you could have very large rearrangements between target sites and off target or two off target sites. So the challenge of being able to control the system is really something that has to be understood in order to be able to push this confidently into products. So what are we doing at NIST? We do not want to stop innovation. Innovation is fantastic in this field. It's pushed us so far forward. But what we at NIST are doing are thinking about what are the measurements that all of the people that are coming up with exciting ideas need to make sure they can innovate with confidence. How do we help with the underlying data? Sometimes you just need that example data set and access to it. And what's the metadata? How do we help with knowing what your process was so you know how to repeat it if it was something that you really got the outcome you wanted, or you know what to avoid if you didn't get the outcome that you wanted. So this is just to introduce you to the genome editing program that I lead at NIST. And we want to support quality and measurements to help translate genome editing into products so that very exciting things that can help people, help society actually get to market. And we want to help by increasing the confidence of the tools people use to understand how they're doing their genome editing and what the outcomes were. So those might be physical measurements, data, or they might be documents. So I just want to give you two more ideas. We have this genome editing consortium, and basically it's bringing together all sorts of partners, academia, industry, government, to address some of the measurements, some of the controls that would be really helpful if we all had a unified something to help. And we're working directly with some innovators, other governments, DARPA, FDA, towards evaluating these new kinds of measurements that are emerging that are needed to help answer some of these questions. And I'm just showing you here that we've been very active in the space trying to help genome editors grapple with standardization. I'll leave you with this. This is our current members for this consortium. And I'll just point out that UCSC is actually a member if you're interested in this and want to try to connect more into this space, as well as Illumina for some of the sequencing applications. And so we're trying to work together to come up with standards, norms to help get these kinds of products, overcome some of these challenges and get these products towards what could be really transformative for society. So I will just leave you here on some hoped and intended impacts. We really want to help accelerate this field together forward faster versus everybody trying to figure out how to get there on their own with this exciting new set of technologies. Thank you very much.
Thank you. We move to Raymond.
Thanks, guys. And thanks, Samantha. And what a great cue up for me. I'm going to do something a little different. I'm going to go ahead and try to share my slides actually on screen with me. And sometimes I'll be on screen, sometimes I'll be off. If you've got me as a speaker, you may want to go ahead and change your view in Zoom meeting to speaker so that I am big and you can see the slides. And if that doesn't work, please jump in and interrupt me. Let me go ahead and let's get started and see how this works out. So, first I wanted to thank the organizers and all the folks at SHCI and Linda in particular, who can't be with us today, but is a friend of mine from a long way back, and my new friends Chris and Manel and Tyler, and especially Jesse and Cisco, who kind of made a lot of things happen whenever I'm distracted and don't answer emails, so thank you especially. Let me jump in now and say what I want to talk about today, anticipating that we would already hear some good technical background on CRISPR. I wanted to talk a little bit about the biohacker piece, and sort of amusingly, I hope, titled this Biohackers, Threat or Menace. And my idea here is people tend to have these preconceived notions about what biohackers are and how that whole thing works. One of the things that I'm going to be talking about is kind of the three classic definitions of biohacker. And there's some overlap between them, but the idea that a biohacker, in my mind, is somebody who is like a computer hacker, but for biology, and less of a movie computer hacker where, you know, it's Mr. Robot out doing all the dastardly stuff, maybe for the good of society, and it's more somebody who's just really interested in the tech and the tools and wanting to advance and find elegant ways to do things. There are a couple of things where the term biohacker has kind of been taken over. People who are doing a lot of work on human performance or self-improvement, and I'm thinking of, in some senses, like the Dave Aspreys of the world, the folks who put butter in coffee and are trying different things. And I think, you know, roughly about 50% of that stuff works, and 50% of that is things that they've just heard from other people that they're trying. But the nice thing is they're trying. And then the third category of biohacker are the folks who are the big self-experimenters. They are the leather jacket clad biopunk biohackers. They're the folks who are doing the DIY genetic experimentation on themselves. Sometimes they're the people who are playing with implants, what are called grinders in the field. Sometimes these are folks who are doing some, you know, really interesting medically valid stuff. More often it's kind of jumped the shark and people are doing things that are dangerous or ill-advised that they're not really qualified to be doing. So, taking those three categories, I'm going to be talking today about the first one, the classic biohackers, and wanted to tell you a little bit kind of about how that has looked for me in my journey. I already got a great intro where, you know, the things to know about me is I went to school way too long and was on the team at Illumina where we invented next generation DNA sequencing, and now I do a lot of work with startups and different companies, partly because of my work with biohackers. The defining graph of my life, and you can't get through any kind of a conference where you talk about biotechnology without talking about that cost of the human genome going down. And that was nice, partly because it has driven so much of the work over the last 20 years, and partly because for me personally, it has been a windfall for some of the things I've gotten to work with and the ability to pay a mortgage in Silicon Valley. The other bit is looking at the CRISPR-Cas9 system, and I don't want to say much about this because I think Samantha's already covered it so well, except I really look at this, and whenever people are like, I don't understand what's going on, you know, you can go back to the idea of it's a bacterial self-defense system to defend against genes and chop up incoming genes, but really it's a Swiss Army knife. It lets us do so many things with gene editing, and I sometimes bring up the qualitative and quantitative difference between what this looks like. So One of the projects that I actually worked on about now almost 9 years ago was a gene editing project where we were using talins, one of the other technologies that Samantha mentioned. And to give you an idea, to do a single edit that we needed to do in production, it took us about 6 months of work to really get it right and try out competing forms of what would be the end result. It took about $20,000 to get through this. Whenever we want to do something like that now, what we do is we get out our phone, we cut and paste a little bit of genetic code. We go to one of the sites where you can order the CRISPR system along with the RNA template that goes with it. You hit add to cart, and instead of 6 months, it's 24 to 48 hours where you get it in FedEx on ice. And instead of $20,000, it's maybe $199, unless if you got a coupon. So this gives you an idea quantitatively and qualitatively how this has really produced a renaissance in being able to, now that we can read, to really now write and manipulate DNA. So enough said about that. There are a number of applications, and I really like the overview. These are some of my favorite ones, and I won't be talking about any of them except incidentally. So now, I was going to say, my personal experience was, I was working for Illumina. We had just put out our first sequencer, and it was all over the news, and we had these ads in the trades, DNA sequencing for everyone. Anybody can do this. This is great. But the truth was, you could do it if you had $1 million to start with, a PhD in a fully equipped lab. That was kind of bothering me because I am a little bit in that tradition of the garage inventor, and I was really wanting to work with other folks who were doing things, and having that high starting cost has always been a problem. What happened is, after playing with some other people in garages, we actually founded a space called BioCurious. And this happened back in 2009. BioCurious is a hackerspace for biotechnology. The mission statement of BioCurious is to make biotech accessible, affordable, and open to everyone. It was founded originally in Sunnyvale. It was actually founded in a garage in Mountain View. With a Kickstarter project. And to give you an idea of kind of how long ago this was in dog years, this was the most successful Kickstarter project for the most amount of money raised ever up to that date, and that was, I believe, $63,000 at the time. So things have kind of moved on. It's now in Santa Clara. It has actually weathered through the pandemic and was able to stay open for some actually really valuable work. It's organized as a not-for-profit, entirely volunteer community lab. It's supported by people paying for periodic classes and monthly memberships to access the space, and is committed to total transparency. The whole lab, and this was the lab on the first day that we opened it up, doesn't have any glassed-in spaces. In fact, there's a blue tape line on the floor that says, if you haven't done your safety training, don't cross this line, but stand on the other side and watch eagerly, and we want to tempt you to come over. Everything is cheap. I show this particular picture because the benches there, the lab benches, were created out of fencing posts and plywood and whiteboard material. And the idea was we were spending everything we could on consumables and repairing old, out-of-date lab equipment, and trying to put all the money in the lab so that people could actually access things they ordinarily wouldn't be able to. People ask what we do. We teach classes, and here's an example class, one of my favorite ones we did. This actually happened over about a week and a half. On the first day, people came to the lab, and we taught them how to build from a kit a PCR machine. And if they didn't know how to solder, we taught them how to solder, and they put it together. And then on the next day, they came back. They isolated their own DNA using the PCR. Here, this is actually one of my students from Singularity University who had come to this class. She is isolating her BRCA1 gene, which is a gene that can show you predisposition for various kinds of cancers. And we teach them then how to use the bioinformatics. So we send it out for sequencing, we get it back. This is old-fashioned, old-style sequencing. With her permission, I'm actually showing the particular trace from her BRCA1 gene, and it turns out, I'm zooming in, if there had been an extra A right between that T and G, then she would have had the bad form. This was the good form, so that's nice news. So this is the kind of thing that we try to do. People who, from a standing start, have never done this before, build their own tools, understand the tools, and do something that is personally meaningful. We try really hard to mentor the next generation of scientists and technologists. As an example, this was a class where we were actually doing CRISPR to make a glowing bacteria. This is sort of the hello world experiment for CRISPR. The interesting thing about this picture, the two instructors are the tall gentleman in the black t-shirt and the short scientist who is clutching her pipetter next to her chest. She had actually come in our lab and said, I already know a lot of this stuff. I want to do something new and different. And so we actually had her teach and mentor one of our classes. The other two guys there are eBay software engineers who were considering changing careers, and one of them did, and that's a fun story. We do a lot of things, and one kind of surprising thing to me, the tech bro problem and the gender balance doesn't happen as much and as badly, and this is from my white male perspective, but we tend to see a lot of women coming and a lot of uptake by women. One of the periodic classes we have is the Saturday morning science class. In that class, on average and sustainably, are 70% of the students who were coming were young girls, and also young boys, but not at 70%. Here's something else that was kind of interesting. Whenever we established the space, one of the things we got was, well, what are your age limits going to be? Are you going to let people in? We actually had some flack from regulatory authorities who were like, if you let anybody under 18 in, we're going to close you down. And interesting stories there. We committed ourselves everywhere possible. We would have no age limits. And we regularly had very complex technical classes with people as young as 5 years old. Here's 5-year-old Harlan making calcium burn red. People would say, do you really let kids go hands-on with blowtorches and different things? Yes, we do. We got a nice award in the first couple of years that we were organized. We were awarded the Fast Company 100, the #4 most innovative education company in the world, which is great because we didn't really think we were about education and we didn't really think we were a company, but we took it and moved on from there. The thing that I had actually done, and what I thought we were going to be when we started, was sort of this clubhouse for garage inventors, and that has been the second big pillar. We do support inventors and we do support entrepreneurs. One of my big questions was, in Silicon Valley, the big thing is how fast, can you get to hello world with a guy and a girl and a laptop in a garage? And that's, how you create your Facebooks. Well, what if what you want to do is not do Facebook, but you want to do a synthetic biology company or cancer diagnostics? And so that was our idea. Could we do that for biotech? And it's really interesting whenever you look at this as a way to leverage people who are trying to play in this space. It's interesting. The people who tend to come in, and I will say this just really quickly, we got a lot of sort of what I would consider a pro-am community. And so there were people who were at schools and they were moonlighting and trying to start their own company, but they didn't want to do it with the school property and with the intellectual property constraints about what people would own. We even have an interesting story where one of Stanford's classes actually ended up being taught at BioCurious, and I was like, there's no way we have equipment that is anywhere close to what matches you guys. And they're like, yeah, but it takes us like 6 months to fill out all the paperwork to do some of the things, and we can do it here in a weekend. And so that was an interesting piece. We had a lot of people who were professionals and moonlighting in jobs where they, again, didn't want to do the work at their employer, but they wanted to do it nights and weekends. It's a nice way to try out a new idea that somebody has where, and that's the idea on this slide to go back, where instead of having to go raise $1 million and buy the capital intensive, fairly expensive equipment, you can come in and use the equipment that is at the space. The other thing that kind of happened is we started to see a lot of outsourcing where instead of people having to do their own work, if they didn't know how or they didn't have the specialty equipment they needed, they could actually send it out, sort of the idea of the Uber or the gig economy, but for research. And we've had that sort of thing for a while with contract research organizations, but some of it got internet enabled and much easier. The other thing is people who come in and use the company, use BioCurious and that particular hackerspace and some of the other community labs, we don't take any IP. We have no conditions except somebody's paying basically money to rent a bench. And that has been really invigorating, and it has actually done well for BioCurious. Several companies that have gone on to do well said, because you guys helped us out at first, both as being a place where we could get started and either fail fast or move on, or a place where we could find our early hires, here is 1% of our equity that we want to donate. And so I actually think that, the idea that these things depend on people paying out of their pocket to rent a bench becomes less likely, because I think the organization will end up doing really well. This attracts a lot of new people, new players, new applications for new markets. We've had over 50 companies come through and out of BioCurious that have been successful in some measure of the word. We have, as of the last time I checked this week, almost $250 million in follow-on funding for those companies. And that's not bad for an all-volunteer space where we don't have a single person paid to run it. Startups and schools have copied the model, and we've now got worldwide biohacker spaces. And one story I love telling is somebody from Johnson & Johnson had come, seen me at a conference, had come and visited BioCurious and said, wow, we should do something like that. Went back to San Diego and started Janssen Labs, which is now a multiple tens of billions of dollar profit center for Johnson & Johnson as sort of a professional lab incubator. We see interesting things, just to show you a couple of the projects that we've seen in the early days of BioCurious. Two of the co-founders of BioCurious had actually created the Open PCR, which was a piece of hardware to do polymerase chain reaction, the copying and amplification reaction for DNA that's at the heart of a lot of things in biotech. It's interesting because they used cheap parts, they used computers, power supplies, 3D, CNC wood, and printed everything out. They sourced heating blocks from China and had their trials and tribulations putting this together, but it's led to another company called Chai Biotechnologies. The OpenPCR is roughly a 20th the cost of a professional PCR machine. And what's interesting about it too is it's a piece of open hardware, meaning that they actually have the parts list and the specs, including the capabilities to CNC this, published, and people take it and then come up with their own hardware standards and republish it. The last I saw, the Open PCR standard was at, I believe, version 5. You can get one now for under $100 or make it yourself at home. An interesting project, somebody came in and said, I'm curious about bioprinting. I would love to do bioprinting. Can you guys get a bioprinter? And we're like, how much do they cost? And at the time, the retail bioprinter was about $1 million. So this guy came in and said, I will teach myself how to make my own. Here they have printed I Heart BioCurious in glowing bacteria, in bacteria with the green fluorescent gene in it. Using a little inkjet cartridge that has been repurposed for that purpose. The goal of their project was to actually be able to print a leaf from scratch. Hasn't quite happened, but they have done some really interesting things and are now working on vascularizing tissues that are used in cell culture and cellular agriculture. One of the neat projects that came out, and this was another community project, and that again surprised me. Somebody would say, I want to learn about this, and then around that nucleus, a group of people would form and say, let's meet every Thursday. These folks wanted to go ahead and make cheese by taking yeast, implanting the genes in, and this was a CRISPR project, and they said, we could do this for, we'll use cow genes, and so that was the first thing that they did. And then they said we could go ahead, we could use human genes, and so they made human milk producing yeast, or human milk protein producing yeast, and that was a little weird, but okay. Then they got weirder. They said if we can do that, what's the weirdest mammal that we can come up with and make cheese from? And so they actually had recently done the narwhal gene, and so they were able to make narwhal cheese. Quick personal note, if you ever get a chance to go to a narwhal cheese tasting party, say no thank you. Moving on, we've seen a lot of things happen, and this is one of my favorite companies. Full disclosure, I'm an advisor for the company. These guys, Miraculous, started out, they did their first sprint prototype project at BioCurious in 2014. They 3D printed a cancer detection microarray reader where you could insert an iPhone in and actually read the spots on this microarray. Moved on, pivoted, and did several things. Today they are making the Miro Canvas, which is a high throughput sample prep device for DNA sequencing, for synthetic biology, for several different things in the research field. And they recently got funding. They're I think they've raised over $65 million and have not a spot in BioCurious, but a very nice spot in Soma in San Francisco. And it's used around the world. So this gives you an idea of some of the things that have happened. Some of the companies that have incubated at BioCurious you would recognize and don't always want to be mentioned because they worry about that association with the idea of hackers and black hat hackers. The big surprise really to me in the whole thing has been the idea that the community is such a big deal. And whenever somebody says, I want to make one of these spaces, help me come up with an equipment list, I'm like, don't do an equipment list. What you want to do is organize a meeting, send out an evite, send out a meetup group, and get people to come and talk about what they're interested in. And then once you've done that every week for 3 months, then you guys work on an equipment list. So the idea, sort of in the words of Breakfast Club, it's sad and demented, it's very geeky but social, and it's a very social space with community, people finding each other, helping on projects. The second most asked question I've gotten about the whole biohacker space thing is, how do people let you do this? There's got to be a law. This has got to be illegal. This has got to be dangerous. And it's been really interesting that people think that. This goes to this idea to me of the 4 horsemen of the bioapocalypse. The folks who are the dangers in this sort of scenario, whenever people come up with spooky things, whenever I look at failed states that are doing weird things in labs, or black labs in not failed states, in very successful states, which have no oversight and no accountability, companies that are doing greedy things without a lot of oversight, or, not to throw other biohackers under the bus, but the idea of the lone biohacker, the guy who is in his mom's basement and everybody says he was so quiet but very nice. These are the things that you really should worry about, because I think what they all share is this lack of transparency. BioCurious, and I actually gave myself the title whenever we started it of Chief Architect, because we planned out the space, is planned specifically to be transparent, where people are working with each other, checking up on each other, where even the storage, there's not a single storage bin there that doesn't have glass doors or clear sides, so that in some ways it's sort of a panopticon. People are able to look and see what's going on. Along the way, whenever we were doing this in the first 3 years, we actually organized a conference with the FBI. We had a huge attendance by Department of Homeland Security and a bunch of acronym agencies, and it was very interesting because the folks there were like, we want to see what's going on and understand. and the folks in the different biohacker spaces were like, we want to show you and we want to share. And it was a sort of a defining moment, because our biggest supporter, actually, in the 7 different levels of government bureaucracy we had to work through to make this happen and open, and that was the very hardest part about doing the first one, was the FBI. The FBI was fantastically helpful and has been really a big cheerleader and has shown us ways that we can do things to work with other agencies. The hardest department has been the fire department, but I think that that's just a thing for small businesses. And it's good that it's worked out, because we're now everywhere. Here's a map. I actually pulled down an updated version of this last night from DIYbio.org. They're currently tracking right now 62 community labs, but another 180 groups and projects and incubators, and on 6 continents, and that actually includes Antarctica now. So it's really interesting to see how this is spread all over the world. I've been to several of the different biohacker labs in my travel. I will be working something commercially or going to give a talk on something professionally, and the biohackers always find me and say, come over and look at what we're doing. Each one of these has a different flavor, a different mix. Some of them are more about education. Some of them are a little more black leather jacket clad. Some of them, the folks in Paris, are very stylish, which I hadn't expected for people who are, after all, geeks at heart, and have had really interesting things going on. So it's been a real privilege to get to see some of those things going on. I guess the last piece I want to say is, as this is spread, what it's doing is it's putting more tools in more hands, and that means more breakthroughs, and this becomes a real democratization for biotechnology, and has really been part of the thing that's feeding into that biotechnology revolution. So that's what I wanted to show you guys. If anybody would like these slides, go ahead and drop me a line at that email, and I'm happy to send them out to individuals. And thank you very much for the time and attention. I'm looking forward to questions and discussion.
That was excellent. That was the best talk we had. Thank you. You're muted, Dr. Ladansky.
I'm sorry. I think I have to modify my grant application. I don't know how this will go with the NIH, but I see the potential to save at least $50,000 over here. David, after you.
Yes. All right. So we're going to open it up for questions. We have some pre-submitted questions. I think maybe what I'll do is open up with the first one of those, and this is questions to each of our speakers. And so this is a question about inhibition or regulation on the CRISPR technology. So the CRISPR's, the capabilities are there, but the restraint seems to be particularly high. So what are your comments about the technology and its more generalized use?
I would jump in just from my perspective on both the biohacker side, seeing what people are doing on that grassroots piece, and on the startup company side. There is a tremendous amount of usage of the technology. A lot of it, and I think that this is good and interesting, is below that clinical regulatory line. People who are wanting to do this and develop it and put things out are trying to find ways, like, can I do it where it's not used in humans, on humans, maybe with humans? Can I do it in food? Can I do it in pets, livestock and animal husbandry? Can I do it in industrial processes? The thing that I think is holding this back from 2 quick perspectives is, whenever somebody does want to do this clinically, there are a number of regulatory hurdles, or at least expensive bases that you have to touch. Part of that is it's really hard to figure out who you've got to talk to and get okays from. Having seen people do some of these experiments in BioCurious and try to move into plant biotechnology and some of that, there are 3 agencies that you've got to work with, and none of them actually, they all try to hand the ball to each other. None of them are the NIST, which does a fantastic job. And by establishing standards, I think helps overcome that problem. The other problem is whenever you're not doing a clinical thing where you're doing a diagnostic or you're trying to do some sort of cell engineering, something like that for therapeutics, you're trying to compete with industrial processes that are at large scales. And so some of the early uses for the technology to make, biofuels, worst idea ever to say, let's do biofuels. Here's something that, sells for less than bottled water and is dangerous to handle and kills off our bacteria as they make it. So finding things where you don't have to scale up to hundreds of thousands of liters a day to be in production is a big deal.
All right, Samantha, any comments on this area?
Yeah, I'll add I agree on the people should have access to use these technologies to be innovative, to be creative, to teach us all things that they're learning because of things they're trying. I don't see any inhibitions there. I think there's a very strong support, and I can speak from the federal government side, working directly with, say, FDA and with NIH, that there's a very, very strong impetus to have really creative, innovative, thoughtful people do those sorts of things. There's definitely a line when you are trying to make something that, as Raymond said, would be in or on somebody. And I work directly with US FDA that would be regulating the gene and cell therapies if you're trying to make a therapeutic for somebody. And that's where people want to know, it can do, so that Swiss Army knife that we heard about, what is it doing? And people are concerned for the, is it safe? Is it safe for somebody? Are we gonna harm somebody? There was a previous time when people were really excited about viral-based gene therapy, and the excitement got a little bit farther ahead than the appetite for being really, 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 really cautious. And, a patient died, and that sort of killed the field. And so people really don't want that to happen again. And so can we make sure that we don't kill this really exciting field because we weren't extra cautious to make sure we understood what we were doing? And so I think that's where it's going. And also the regulators that would actually be from the US federal government side, like required to regulate this, they're learning as we go because the technology is moving so quickly. So they're trying to, figure out what the rules are, what the concerns are as things go along. And that just creates challenges to know how to move forward. So.
I think both points are very, very valid, but what comes to me after listening to both Samantha and Raymond answer, and sort of fusing with the conference that we have yesterday, we're talking about the, brain software interfaces. I kind of wonder, what if I'm in the unit and I have somebody who didn't wake up from coma, and somebody looks what Raymond is telling can come and say, hey, I have this gene which actually may help because I read this case report. And I actually can bring and say, why won't we try it? Maybe I'm a little bit pushing, although yesterday I was told that in 2 years, a lot of change in the human brain interface. What do we do then? Like, what if the people start trying, treat this actively? And what Samantha is saying is true. There was adenoviral trials, which kind of went sort of not the way as expected and killed the field for 15 years plus. Mm-hmm. So how you balance that? And, this is US. What about the other countries when, this regulation may be a little bit different, but then those people are sort of, ethical or unethically getting ahead of us? How you balance all those things?
Yeah, I think the balancing is challenging, but as you just pointed out, there is awareness, at least on the US side, that at least with the CRISPR technology, a lot of the innovation in the CRISPR technology came out of NIH grants that went to researchers and helped with, inventing and fostering and institutions. So there is a heightened awareness of not having that sort of IP, that innovation where the US is leading, not to have leadership there and sort of miss the boat on something that we could have really, gotten a lot of benefit to our population out of, and not have somebody else own with IP or even global IP, something that we're now restricted from. I think the idea of like that biohacking, it's not something that's very familiar in my world, but if somebody has an idea that they have an option to go somewhere like that and say, can we try there? Because I think it'd be extremely challenging for somebody that isn't recognized as somebody that does this in the field to like try to say we should, do this at a university. But to have options like that, I think that's where we might be able to come up with new paradigms. We need new paradigms for exciting technology, to reach its potential.
And sort of leading to Raymond on that, again, I'm writing the grant, and I do know that the genomic part, like 60%, is so grossly overpriced. And then I'm hearing about the PCR machine, which, cost, I get my stuff on the eBay occasionally or on the Amazon, which is kind of 1/10 of the regular prices. And I'm hearing, I can get it even by 1/10 of that price. So I'm like, maybe I should just write a grant and move everything to that facility because it's cheaper and I can do more and faster. So I can absolutely see the appeal. Like Raymond, how do you see this enthusiasm blowing? Because I'm sure that people are coming back and forth.
Let me walk it back a little bit, because I don't want to oversell the reality of especially a biohacker space or a community lab, or there are different ways to look at it. Shared equipment incubators is really the more formal model, but there are I think there are huge advantages for certain situations and at certain stages, and there are huge drawbacks. Part of it, think of any time you've ever had roommates and shared a kitchen. Everything's always messy. You can never find anything, and it's exactly those sort of things. But you're in the middle of your tissue culture trial and trying to figure out, and also, you've run out of the reagent that you need. How did you guys not order this whenever you used up the last thing in the jar? And we do have those things. The people who've been really successful with this are the people who wanted to try something out quickly, and instead of having to quit their jobs and raise a bunch of money to try something, then it was like, they were like, okay, we did it. And 80% of all those things just fail, or people pivot and try different things. The ones who have been successful, that was one of our problems commercially with BioCurious and some of the other hacker spaces that have been incubators. The people who are really good at what they're doing either fail quickly and are done, or they succeed quickly and then they move up and graduate out to something. So you don't have a lot of long-term tenants. We do have long-term tenant hobbyists and long-term tenant people who do sort of services for other people, and that's really interesting. And the educational piece is evergreen. We also have our own problems with regulatory issues. There's one thing that was sort of an infamous incident in BioCurious history. We had a company that had we had come up with our own genetic engineering program for plants, which is actually fairly hard to do, fairly sophisticated. And we were using agrobacterium, and we were using a little DIY gene gun. And I think are still the only, but maybe one of two biohacker spaces that had been able to do both of those. The company that was wanting to make glowing plants started all their work there, and I thought that was really interesting work. But what they wanted to do was raise money on Kickstarter and send the seeds out to all of the backers before they had any kind of regulatory approval. And they were doing it to be deliberately provocative. And we had to stop them and say, no, we, even though this looks like wild and woolly and the West, we have this set up, and we only are able to receive plasmids because we have certain standards. We have material transfer agreements. We do and don't allow people to take things out of the lab into the environment or just home. And so none of that's allowed. The stuff that you guys are working on has to stay here until you have a proper facility and you can do an MTA, a material transfer agreement. And so after that, I found myself and the organization being sued by a group that wanted to stop us from allowing people to do that genetic engineering work in the lab and make sure that it didn't get out, and the group that had stuff in the lab that they wanted to take out before they actually had money to build a space that was even BSL-2 compatible. So it's, it becomes interesting whenever you actually are at that nitty gritty and trying to get things done and release them.
Of note, this really brings us to our next question, which I'll post in chat and also speak to. Who should regulate CRISPR use and in what technical domains? And so this is exactly the topic that both Raymond and Samantha were bringing up. So clearly we have uses that are both broadly used and others that need deep care and oversight. So what are your thoughts here? Who should regulate this?
This falls within kind of the infrastructure, at least in the US, that already exists. If it's a human therapy, there's a unit that does that. The challenge is, as Raymond was saying, is that agriculture products, the infrastructure is like co-regulation. Like it depends on like partly like where it was manufactured and then like where is it going to, and like, even within the government, that whole thing confuses me. Like who even I should be talking to about like measurements and standards to help all of this. So sometimes there are overlapping jurisdictions, and I think it's mapping onto some of these things. But I think some of it becomes more provocative when you think about differences in cultures where the US's regulation of, say, agriculture products is very different than the way that the UK views. Not the UK, the EU views engineering of agriculture products. Like those are all GMOs, and they are not they're not thought of kindly there in terms of their GMOs, versus in the US, depending on what you're doing and if it was a natural variant, it is either regulated or not regulated. And I think there are opportunities for engagement, because the regulators are trying to figure out what's reasonable and, developing as things go along. But I think it can be challenging to figure out like who regulates that. I think there's sort of like a 3 for like agriculture, and then like a 1 for the human therapeutics. But like, what if you're engineering an animal to do something else and it's not like food, and then, it's a little bit more challenging. But I think this also comes back to the question of should there be any form of regulation? And I think some people say no, things shouldn't be, and other people say yes, there should. And I just want to put out there, would you feel comfortable if you, went to the doctor and there was no, norm, standards, regulation at all? And I think you'd be uncomfortable with that. And so, we still want to, if we wouldn't feel comfortable eating it, consuming it without knowing that there was some level of quality or safety applied to it, then we can view that in the same way as well as should or shouldn't. But then we might engineer people at some point. Are we GMOs at that point? I don't know.
That idea of, your doctor is prescribing a therapy and it's like, well, what's the testing? Well, it got good reviews on Amazon. People seem, it's, 4.5 stars. So that doesn't seem like a great idea. I love my FDA when it's doing great things for me. I love APHIS and all of the other, multiple letter agencies that keep pests and things from multiplying. One of the things that was super interesting to me, so it's sort of a classic Silicon Valley entrepreneur thing to gripe about regulation and regulatory overhead and bureaucracy. But the hardest thing we had to do whenever we started BioCurious was figure out what laws we were breaking. And because, we had the equipment, people kind of knew what they were doing. We only had 2 scientists in the group. We had a philosopher, 2 economists, and an MBA and an electrical engineer. So that gives you an idea how people can come together on these. But we sat around my kitchen table for a year, once or twice a week, basically researching what are the regulatory things. There aren't any. Like if you want to do genetic engineering at home, you can just do that. If you want to make a thing and make a seed, you can do that. If you cause a problem with it, if it spreads across state lines, or if it's a pest, then it's a liability and then you can be sued. So what that means is professional organizations have certain standards, and I want to talk about that in a second, and corporations have liabilities, and they have millions and billions of dollars invested, so they're careful for the most part about what they're doing, because they're trying to do something and they get benefits, but they also get the risks, and it's attributable to them. Whenever you look at classically how this field has been regulated, especially with regard to recombinant DNA, it was really interesting, and anybody who is interested in the history of technology ought to read about the Asilomar conference, right? The idea where basically half of the geneticists who were working on recombinant DNA and microbes said, this is great, let's just push it to the envelope, and the other half were like, oh my God, no, we could actually really cause some problems. And so they got together and said, what are we going to do? And the answer was, let's self-regulate. Let's come up with our own guiding principles and do things, because we've got a lot, basically if the government gets in on it, nobody's going to like it. We're going to have a hard time. We don't want top-down regulation. And so far that has worked. What may be interesting is the technology has gone so far that may be breaking now, because you don't have to have gone through a professional PhD program and be instilled with a certain idea about the gravitas and some of these things yourself to do this work. You can actually be a hobbyist and experimenter and get all of the materials and all the things. And this isn't like, people always say, well, we regulate atomic weapons. We will never regulate this like we regulate anything internationally with atomics, because there is no hard material to get. You can get all the stuff in your backyard, or you can make it from scratch. That makes it really hard to contain. So self-regulation will continue to be a thing. The other thing is the advance of technology puts more tools in more hands so that smaller groups with less resources can do more amazing things. And I really stay on top of the field for a lot of reasons. And I remember being amazed about 3 years ago whenever I heard that a 5th grade classroom, with some help, had actually constructed their own virus to show that they could do it, did a synthetic biology group science project. And I was just like, oh. So it happens really fast. Smaller and smaller groups with less resources can do more, and they don't have that liability hold back. And so, how do you make that work? I think self-regulation is good. I think liability is good. I think that there are some cracks where that doesn't work. I like biohacking. I think it leads to good results, even some of the things that I'm a little uncomfortable with. George Church, who is, of course, all over this field, the Wyss Institute at Harvard, a geneticist and molecular biologist, has proposed that maybe we need a driver's license for molecular biology, for biohacking. And we actually had kind of done some of that at BioCurious, where, to walk into the lab, you can look at anything, but you can't touch. Before you can actually be accepted as a member and get a key, you had to go through some fairly extensive safety training, and before you can use different pieces of equipment, you have to either demonstrate you can use it or go through the training for it. Not every space has the resources to do things in that much of a bit, but the idea of a driver's license is probably good and analogous for this sort of thing. The last thing, and I'm sorry I give long answers, internationally it's really interesting to look at what's going on. Europe tends to operate under the precautionary principle. If you haven't proven that it's okay, it's not okay. That's why GMOs are still not a big thing. The governments of Europe love GMOs and would like to have more plants in the income with hardier plants and more pest-resistant plants, but the people don't at this point. In the US, it is pretty much, well, if you haven't proven that it's harmful, then just go. And so we have very, very little regulation. Like in Germany, you have to have a license or a registration for a PCR machine, which I thought was really kind of funny. And here you can buy one off of eBay and do what you want. In China, it's very interesting to see what's going on with, for instance, BGI, which used to be the Beijing Genomics Institute, but is now kind of in turns a government institute, a not-for-profit institute, and very much a for-profit company that has kicked out a stock offering. And they kind of retreat into whichever one of those they need to do what they want to do. And they have more bioinformaticists there than pretty much the rest of the world combined. The Chinese government spends more money on genetic engineering research programs, in particular with GMO, definitely than the rest of the world combined, corporate, government, and academic. And they have a really different standard, and I won't say it's unethical, because I don't think it is, and I think some of that is really just like Asia bashing, but it's a different standard of what is okay to just go ahead and do, and how do we do that. And whenever you look at this with these world-spanning, world-affecting technologies, where you can make something in one corner that will affect everybody, that's in some ways really heartening that different people can try different experiments, and really scary too. The one thing that I think keeps me up at night is I don't see that there's not a policy solution that just works for any of that.
I'm going to extend this, and then I'm going to get back to some of the questions that have come up in the chat. So let me get this one sent. Oh my goodness. So let's consider some examples here. So, and again, this is back to this idea of different levels of regulation. So E. coli that glows green. So that's an example. What ought to be the regulation? And interestingly, how much of a driver's license do we really need to instrument E. coli? We had a question that was pre-submitted about domestic animal manipulation. And so what are the regulatory issues here? How much of a driver's license do you actually need? And then I'm gonna push that a little bit. And what happens when we actually leave even the national boundary, and we're now considering effects that may have global implications? So are there downsides to eliminating mosquitoes from the planet? And by the way, we can do that with CRISPR technology. Who controls the work? And back to your comment about the Asilomar agreement, where is that? Where is the self-regulation that now may need to be handled at the global level? So big set of questions all around regulation, from E. coli up through really the globe, the planet we live on.
Maybe I can take a stab at some of
There you go.
So for domestic animals, I think this is also one of those lines where, depending on what you're doing with them, it might be USDA, it might be FDA. Particularly if it's for some sort of biotechnology, that's definitely FDA, Center for Veterinary Medicine. And if it's for food, I think it might be some of both FDA, Center for Veterinary Medicine, and USDA. So it has a lot to do with what your final intended use of that animal, that domestic animal is, whether it's food or whether it's like to make a secondary product. And again, it has to go back to like the safety of it, because these are the same organizations that regulate all the food, whether it's engineered or not. So that's where some of that lies, but it's still being worked out, because it's very early days of somebody getting to the point of saying, I would like this domestic animal product that's engineered to be into the process. Because while genome editing and CRISPR technologies are so widely accessible in a lab setting, they can be challenging to make the product that you want to make, and have that livestock, come to term and be what you need it to be. So there's a lot of challenges just in people applying those to complex products still. So we're just at the beginning of these things really pushing into the regulatory space, and they're learning as they go, as the different use cases start coming to them. I will also take the self-regulation thing. So several years ago, there was actually at a Cold Spring Harbor meeting on genome engineering, a discussion basically saying we need to come to some standards and norms about behavior. And that was sort of an informal discussion, and it sort of, has started coming out a little bit more in terms of my world of actually, standards on the how-to side, as to make sure that you're getting something that you want and you're interpreting the data correctly. But there's been this other conversation happening between the U.S. National Institute of Sciences and the UK, the National Institute, the Royal Society, and China's National Institute of Sciences around human gene editing. And that would range everything from therapeutics, things that I think globally, generally, everybody says this would be a good thing to do if we can help people, all the way to enhancement. What is our general perspective on people that aren't sick, that might want to be able to enhance themselves with things like CRISPR technologies? And can we have some global discussions? Can we come to some agreements? And the answer is there isn't going to be any sort of global agreement. It will be respect and hearing each other and understanding, but that within each country or within each region, there are probably going to be some norms. The US has put out a very clear set of standards, expectations, operating procedures as to what is able to be done with federal funds and what's not able to be done with federal funds, and then just guiding principles of whether they think you should do this even if you have your own private funds. But the FDA, for example, has stated unequivocally that they will not consider approving or reviewing heritable human genome editing samples where you might have edited an embryo or a person such that it would be heritable to the next generation. So that's a US policy side. It's not necessarily the same in other countries, although there has not yet been a country that says we're okay with engineering another generation of people. But there are different levels. In the UK, you're allowed to do human embryo editing as long as you terminate those cells by, I believe it's 14 days. So you can do research on early human development. That's not the case on the US side, and it's unclear in other regions at times what they're okay with. Coming back to what Raymond was saying, like what's the appetite for how far you can go. But within the technical community, there's a sense of the technical community coming together like a Silomar to say, what do we feel we're comfortable with? What do we think is ready? And I think that is bounded by a lot of things I was talking about, where how much do we know about what's actually happening in the cells when we apply these tools, and how comfortable are we with applying them at a level that's even more important or maybe heritable to the next generation? Are we really comfortable with even thinking about moving forward with that before the government tells us whether we can do it or not? And some of those discussions have definitely been happening so far in the field.
Okay, I'm going to let us all relax a little bit. I'm going to ask an easy one, and then I'm going to, of course, follow it up with the big question that you both know I'm going to ask. So in terms of naming of the activities, biohacking has many meanings, and Raymond, you talked about this earlier on. Are we too late to make a naming shift to define the different domains of amateur to maybe criminal modification of biomaterial? Are we too late, or are we just using this one term biohacking and we have to accept all of its different uses?
Yeah, I think terminology gets out of control of people. It's just so slippery, and you end up kind of stuck with things. And then you end up with alternative, maybe more politically correct things, which is why you'll hear me refer to our spot as a biohacker space half the time, and a community lab, which has got a really different word charge the other half of the time. I think that what we're too late to put that word genie back in the bottle. And part of it is, even if you really tried to and you started to come up with an ontology of what are acceptable activities that would fall under which umbrellas, we are just never, ever, ever going to have a black and white on the range of activities that you can do, and then kind of putting that with the other dimension, or getting individuals, societies, different groups, regulatory or social, religious, to agree on what's okay and what's not okay. And as a recovering engineer, right, I used to think there was always two options. There was the right way to do something, and then there were all the other wrong and less efficient ways. And whenever you come into this field, you really get a chance to see there are a lot of pieces of gray area. There are exceptions to every definition. I can come up with something that will curl your toenails about, here you would say this might be okay in every possibility, or this would be not enhancement, and that, and come up with something with a counterexample where you'd be like, oh my God, we shouldn't allow that. And we could do it and actually split our room right down the middle. There would be things people would love and hate. Samantha made that really great point. There will never, ever be an international consensus on this. And in some ways that's a strength, because that means we've got about 183 countries doing different experiments and trying things to see what works best. And as people can move around and do medical tourism, and things are evidence-based, that's a great outcome. As places where you could have dark corners of the network and the globe where bad things happen, that's not so great. Different standards, of course, for human therapeutics, for environmental spread, for other interesting things, and it's going to be a sliding scale. But again, we will never get people to agree on what's okay and what's not okay, so we will for sure never have words that we can define. And people will always mean different things, they'll be differently charged.
Okay, I'm going to offer this, and let's see how far we get with it. If we in the US decide not to modify heritable humans, are we then giving this sort of modification to the domain of others? Will we modify the human state? And does this create a new definition of humanity and human-like beings?
Let me jump on that grenade. I love to, Samantha had brought up the FDA decision to not allow human germline genetic engineering, and it's kind of interesting. And this is again, this kind of goes back to that last question, like, how do you regulate, where do you do it, what goes on? Human cloning is actually illegal in California. It's not illegal in Nevada, which is kind of funny. And there are no human cloning programs that I know of in Las Vegas, but I wouldn't be surprised. There are human cloning programs that I know of where people are attempting or saying that they're trying to attempt this. There are human germline engineering programs that I know of internationally where people are doing this. The reason the FDA is not doing human germline stuff now is because in a funding bill for highway and defense, somebody slipped in an amendment that basically said the FDA may not consider applications for review of anything that involves human germline engineering. And it's put in a poison pill. It had nothing to do with it. It just kind of got it in there. And so without somebody passing a law that basically rescinds that, there is no way that anybody can legally do something and put it through the FDA. Could you still do it at home? Could you do this and just not make a product, or just not do different things? Another thing we had brought up, the idea of dog engineering. That's a thing where, again, if you're productizing it, somebody should review it. Somebody should do some things. But there's a guy in Mississippi who's actually working on hip dysplasia in Dalmatians, and contacted the FDA and said, I want to do this for my own dog breeding. Is that okay? And they're like, as long as you don't sell the dogs. He's like, I'm not trying to make money off of this. I'm going to do it and then show people how to do it, and they can do their own. I'm going to give it away. And the FDA kind of freaked out about that. And you can see it's like all these things, because they are cheaper and easier to do, because this is not $1 million project for him. This is like a $24,000 project. He can go ahead and make this attempt where people couldn't do it before and wouldn't be restrained by these things. So I don't think it's going to matter what we decide about modifying hereditable human beings. We're going to see it happen. I think that argument is always, if we don't do it, someone else will. I think that there's a moral problem with that, because people then use that as a shield. So let's start doing it. That doesn't mean I think you should start doing it and doing it wrong. How do you actually do it? But, I think it is a fairly powerful argument. If someone else is doing it, can we do this well? One of the things, and this I think is especially true for U.S.-China regulation or relations and commercially, people say the biggest export of the U.S. to China right now is regulations. It's like, if you're not doing it right, we're not going to be your market. I do think we will see this. We will see both, somatic engineering for disease. We'll see germline engineering for disease, and some things that will go across that slippery slope from disease to enhancement. Some of that has already happened. I think some more of that has already happened that people aren't talking about because of the poor reception that Dr. Hay got whenever he did his work in 2018, and perhaps by cutting a couple of corners there. But I think it's happening now, and we're going to see it happen in so many different ways, and not necessarily in the ways that we would assume, predict, or hope for. We're going to have to figure out, how are we going to deal with it. I think it's a done deal. I guess that's whenever I put on my prophet hat and get out my crystal ball, I think we're really looking in the rearview mirror at this point. I think all these things are happening, and maybe we can come up with some frameworks that help us corral it a little bit, but it's definitely a case of not being able to stuff the genie back into a kind of too small bottle.
All right, Samantha, did you have any last words on this grenade as it was described?
I just want to point out that we have to think about things in context. So, when you get on an airplane and you fly, you're damaging the level of radiation, you're damaging your DNA. These are things that we expect. You could damage DNA anywhere, including, in your germ cells that might contribute to the next generation, what you choose to eat, the way that you live your life, the environment. These things are happening in an uncontrolled way, and you impact your decisions. If you are, a parent and you have a genetically related child, impact, what you pass on. So the idea that the decisions we make are heritable to the next generation, is something to, think about as a normal part of it. And I think it depends on the purpose, and that's where different countries are kind of evaluating, is this the only way to do this? And I'll go back to also saying that the application of trying to do germline engineering is one of the more complex applications, because you need the materials to do it. So you either need to in situ engineer in the ovaries and testis, or you need to take those things out and then do in vitro and put that fertilized embryo back in. So this is probably the most non-trivial application of these technologies. And so I think you need a compelling use case. And I think in the US, there's a less compelling use case for doing that so far. Where I think that range is definitely starting to happen is, it will be hard to know if these technologies are safe if you don't have the ability to do that research in an embryo to know if it's safe. And whether you think it should be done or not, whether, putting all ethics aside of whether, what one's sensibilities are, you would need the opportunity to do that work in that system because embryos function differently and their regulation is very different than what you think of as a fully developed adult cell. And they don't like having their DNA broken. They've got additional controls because it's a very detrimental situation to inherit that or potentially have all the cells in your body have this particular damage. So that landscape is not as well understood. And then it goes back to, should one be doing that research to understand that? And like Raymond said, some places are saying, yeah, we should be doing this because there's a benefit, there's a utility to our nationals in our country or our region, that they could have benefit from this. And other places are saying we're not comfortable with this for whatever reason. I agree from my perspective. I think it's happening and it's going to go this way. How much it happens or will happen, I'm not sure, because this is also going to be expensive. So like the reality of the situation is challenging. It requires specialization, and it's not going to be a trivial in cost. The cost is not a limitation for some people if it's available. And so I think there will be markets that may want something like this, and there may be regions that will allow this, but is it socially acceptable? A lot of times things that are not socially acceptable, even if they can be done, don't really make their way into society. So I think that'll be the question.
All right. Thank you both. And I truly appreciate your candid and well-reasoned answers to each of these. I think I'll turn this back over to our organizers.
Thank you very much, David. I think one of the purposes that we opened the organization is to open the eyes to the people that we work with, that some of the questions that they think is 20 years ahead, they're around the corner. And I think that session clearly demonstrate that. The second thing, we always struggle. We always get the speakers, or we try to get a speaker which are slightly misaligned, and it's kind of fascinating to look both sides right here. And I think also what you very nicely demonstrated, that the people can have a discourse without being insultive, which unfortunately in the current world seems to sometimes be a big issue. So we can disagree in the civil way and move the needle the way that we are all happy. So thank you very much for that. This session is coming to close because it's our final session of this year edition of the Innovation Frontiers. I need to mention all the organizers because I'm just a person who opens the Zoom and close. But one of the biggest kudos should go to Linda, who cannot be here. Linda, I met Linda around 6 months ago. I was absolutely exhilarated by her knowledge and ethical levels, and also how kind she was. Unfortunately, Linda is going through some health problems. United States has one of the best medical care system. This on the individual level, we can discuss on the social level. And I think her recovery is simply inevitable. So kudos to Linda. Again, we should also wish her speedy recovery. The other kudos goes to Tyler. Tyler very nicely stepped in when the things fall through the crack. And he filled all the holes and everything went very, very smoothly. I'd like to also thank profoundly Dr. Kamps for being my partners in crime. And even in the face of sometimes difficulties organizing the things, we keep smiling at each other and pretend that the problem doesn't exist or it's absolutely workable, or whichever way it will be fine. The last three people who are, like, one of them is Cisco. Cisco is my colleague. We used to work together at the University of Pennsylvania. Now he moved to Missouri and he's very interested in society. Jessie is our administrative assistant, which is understatement of her role. And Mareva is the person who does the social media. Also social media was done by the team from the University of California, Santa Cruz. So kudos goes to those people as well. And closing our session today and thanking all of you for the contribution. I hope we're going to see in one form or the other in October 2022. Also, we'll see who was right about when the pandemic will be over. Thank you very much. Have a good evening.